will be talking to you. Oh, sorry. Today I'll be talking to you about a general overview of what I've been working throughout my PhD and going more towards the end with the work that I'm doing currently. All this work has been tightly done with Thomas Kjørbo, who has been my supervisor throughout my PhD. And I've mainly been working on phagotrophic flagellates, so I hope to inform you more about these organisms throughout my talk. So first, I would like to start the talk with this image. It's a recycled image from a colleague that she generated with AI. You can imagine that the prompt had something to do with flagellates, but when I saw this image generated, I could not recognize almost a single flagellate. I guess with this, I want to point out how flagellates in general are just put into this like black box and not really known, especially for the general public. And yeah, you can find some flagellates that may be a bit more close to what the artificial intelligence may imagine they are. But in general, flagellates are just a single cell organism that have one or two, sometimes four or up to 8 flagella or more. But the general, you would see 1 or 2 flagella that they bear. But you can see here how there's a great diversity of morphology and different structures that they can have added to be able to do their perform their vital functions. I focus mainly on phagotrophic flagellates, meaning that these organisms have to prey on other organisms to be able to feed. And they engulf the mainly bacteria or phytoplankton through phagocytosis. Phagotrophic flagellates are also very diverse phylogenetically. You can find them across all the eukaryotic tree of life that you have here represented. Sorry, I'm going to change my pointer. Oh, I cannot change my pointer. Yeah, there it is. I didn't do that in the beginning. So you can see how they're present across the whole eukaryotic tree of life. Here highlighted are the groups that have phagotrophic flagellates represented within these groups. So you can see that they're very abundant and yes, but mainly only if studies have focused on a few of these groups and many remain quite unknown. And to add to that, you also can see how within the recent 10 years, this is a timeline of different studies and description of new lineages and species. So we are adding more to the unknown, but also with the descriptions of these new species, we're able to also construct better an idea of the phylogenetic relation between these groups and to understand more the tree of life of eukaryotic organisms. But yeah, many of these groups are also remain just described, but their function or their way of feeding remains unstudied. So they are very ecological significant, the phagotrophic flagellates, mainly because they play an important role in the microbial loop. So basically, they're feeding on the bacteria that are feeding on the dissolved organic matter that is released by the main food chain in the marines or aquatic ecosystems. So all this dissolved organic matter is consumed by these bacteria, and then the phagotrophic, mainly phagotrophic flagellates are going to be feeding on this bacteria, and at the same time are predated by bigger zooplankton, which is returning this energy that otherwise would be lost back into the main food chain. But how do they feed on bacteria and phytoplankton? Because being of such small scale, because we're talking about organisms that are around 2 to 10 microns of cell size diameter, well, being in the low Reynolds number regime where viscosity governs and there's no inertia, well, they have to generate these feeding flows in which they're able to capture the particles that are embedded in these feeding flows that are then drawn towards them, and then they can handle this prey to be able to phagocytize them. So this is hence the title of this talk. We're going to be talking about mainly the generation, the different ways in which these organisms generate these flows. And these flows are generated by their flagella. So this is you'll see this is a cross-section view of the flagella, and across the whole tree of life, where I've shown you all the different groups, they all share this ultrastructure, but they all use it in different ways. So I'm going to hopefully be able to explain to you some of the highlights of different manners of being able to generate these feeding flows to be able to successfully feed on bacteria and phytoplankton. In this talk, I'm going to be first I'm going to be dividing it into hairy flagella and flagella with veins. So I'm going to be always jumping from one type to another. So I'm not going to focus much on species names and all other details. If you want to know more about that, you can ask about it later. But I'm going to be referring to one type of flagella equipped with hairs or mastigonemes, these stiff little appendages that protrude from the flagellum, or this configuration over here that you can see, which is the cell that bears like this pocket or this ventral groove that they call, and the flagellum is equipped with this thin vein. This type of morphology is characteristic to this group that we call typical excavate. So sometimes I'll be mentioning typical excavate morphology, this is what I'm referring to, the cell having this ventral groove or this pocket, and that's equipped with this, and the flagellum is equipped with this vein. It is known, and you can maybe look up this study that I cite here, that for more information, but it is known that hairs that are equipped with mastigonemes or that are hairy flagella, they generate they're able to generate a bigger force towards to be able to generate their feeding current, because the hairs act like this sheet in which the thrust is bigger at the crest of the curvy flagellum that is beating actively to be able to generate this feed, and then they're able to generate a feeding floor that is towards the cell body, to the contrary to a naked flagellum that would be generating a flow that is outwards. So basically, they inverse the flow direction. Flagella with veins, it has been studied, for example, that flagellates also bear flagella with veins, but the configuration I have talked to you about, the typical excavate, had not been studied before. You have many ways of producing feeding flows, so there is not a single one. That the diversity is quite astonishing, and just looking at a few species, you can see that the configuration of the flagella changes, the position of the cell, and the direction that they generate the flows can be very varied amongst these different groups. So you can have, for example, here you have the hairy flagella group, which you have this organism that attaches to the surface with one posterior flagellum, and the anterior flagellum curls around forwards to be able to generate this flow, which is indicated by the moving arrows. On the other side, you have the flagella equipped with veins, or from the excavata group, or the typical excavates. And these ones have veins that are maybe towards outside, here in yellow, for example, or towards inside the groove, like here on top. And then they may be attached to the surface by different manners, or else just skidding along the surface, like this organism over here. But this is how they're all generating this feeding flows to be able to capture their prey. We have measured these clearance rate, this flow, or basically the clearance rates, and that would be the maximum volume that they're able to scan through their capturing area. We've measured them using neutrally buoyant microparticles, and we were tracking them, manually tracking them, that passing through the capture area. And you can see here how the beads go. This is one of the organisms with the vein flagellum, a typical excavate, and you see how it's going through. So by getting the speed, and then we were able to also measure the capturing area, or basically the area of the pocket or the ventral groove that they have, then we were able to acquire the amount of volume that they're able to scan. And then the same process was here. This is just a graph representing the tracks or the beads that were captured in green and the ones that were not captured in red. And basically that's how we were able to define the capturing area, and then also be able to measure the potential clearance rates. And our results were more or less what we were expecting from what is in the literature. And they're able, despite the different ways of generating these feeding currents, they're all able to scan through an order of 10 to the 6 times their cell volume a day, which is quite incredible. But they all, regardless of how they're generating this feeding current, they're able to scan successfully great amounts of water to be able to capture their prey. In the case of the hairy flagellum, we were able to estimate the force that this flagella is generating, right, for these different cases of ways of producing this feeding current. We used a point force model that was, starting with the fact that in a low Reynolds number regime, the Stokes equations that govern are the ones of the momentum conservation and the mass conservation. And then we have Blake that generated, he derived an equation of a flow generated by a point force that was close to a wall. And from that equation, one of my colleagues was able to derive an equation that was calculating the flow that was passing through a clearance disk. So this theoretical disk where all the particles are passing through that will be potential food. And he was able to derive this equation here that was, Q would be the clearance rate. And then you have D, which is the distance of the clearance disk from the wall. You have F, that is the force that the flagellum or the point force is generating. A is the radius of the disk, and H is the height of the force that's generating this flow. And obviously, in our case, we already have Q because we were able to obtain it through our experimental observations. So from there, we were able to just grab all the parameters that we needed to be able to calculate the forces generated by this hairy flagellum. We produced these theoretical feeding flows, which were compared to our experimental observations to be able to make sure that we were getting something with more or less sense. And then we were able to obtain forces, estimated forces that these flagella are producing to be able to clear such great amounts of water. And they were all around the order of 5 to 12 piconewtons, which is quite in agreement with other estimates that other studies have obtained with organisms of similar size. And here, you can see the different species that we use, the experimental manual tracking, the theoretical flows, and then the estimations of forces that we obtain. Oops. Is it frozen?
Try to move again.
I don't know why it's frozen. I'm gonna have to close it, I think, because it's...
Okay.
Oh, no. No, perfect. Okay, so now going towards the other case. So we talked about now estimating the forces that were that the hairy flagella were producing. Now we're going to the case of the flagella with veins. So the typical excavate morphology. So we had different species, and as I mentioned, these species have a different orientation of veins. And like here, we have the vein that was towards the ventral groove. This is a view of the cross section of the cell, and you can imagine the flagellum beating inside the groove with a vein towards the ventral groove, one that's towards outside of the cell, and then even we had some cases with two veins on, well, one vein on each side, so two veins in total. We're actually able to observe with the microscope the veins, which had not been done before, and we're able to confirm the direction of the vein while they are beating and clearing great amounts of water. And then we're also saw that there was possibilities of different directions of this ventral groove, whether it was facing up from the surface or facing down. So we could see that there was many different possibilities and configurations and to be able to produce these feeding currents. So a colleague of mine that is able to, he's really good at doing computation fluid dynamics. He basically generated this basic model of this excavate, or this typical excavate morphology. So the cell with the pocket and the flagellum with a vein. And then he was basically able to like play around with it when generating different configurations of different settings of these morphological structures. So by playing around a bit, he was able to get the really interesting results. But I want to focus on for the fact that the function of the vein that he found is mainly, if you see at this graph in the bottom, you can see that there is the naked flagellum is starting here on this point, and then you have the clearance rate on the y-axis and the power needed to be able to generate this clearance rate. And you can see that by increasing the beating frequency, you need much more power to be able to generate the same clearance rate as a naked flagellum with a vein. But that would need much less power to be able to generate this clearance rate. So basically, you have that a vein flagellum is much more effective and will generate much more of a clearance rate using the same power as a naked flagellum. So the vein is basically increasing the efficiency, but then what is the role of the groove? And what we're able to see is that the groove, and this is on this graph on the top, you have the clearance rate again. On the y-axis and on the x-axis, you have the width. And this is the width of the vein, sorry. So then by increasing the width of a vein, you will always have higher clearance rates with a groove. And that's because the groove is actually giving more direction to the flow, and the resulting net flow will be higher. So these two feeding structures have co-evolved and are very important for them to work together, because yes, a vein flagellum will not be as efficient as without a groove, and a groove will not make much sense with a naked flagellum. So the typical excavates that have these feeding structures have been able to have two feeding structures that work very well together. Now that I've talked about how the flows are generated and the different cases of the hairy flagellum and the vein flagella, I want to just go a bit over how they capture their prey. Especially because there's many different ways, but I wanted to point it out how some species are able to capture their prey while keeping the prey within with this synchronized the synchronized beating between two flagella. So we have this species, for example, that is attached to the surface, and then it has a beating flagellum that is the red one that is generating this feeding flow. And as soon as the particle arrives, it is able to change its beating kinematics, so the behavior changed totally. And then the anterior flagellum, that what had been resting until then, becomes very active, and it's able to keep the particle or the food within these two flagella, never establishing contact until it decides to phagocytize it. So here is an example. The prey arrives, it's changed its behavior, and it's never establishing contact. So it's like within the flow of these two synchronized flagella until it decides to eventually eat it, start phagocytizing it. So in the other case, though, we have the excavates, so the flagella with veins, and they're more or less continuously generating a feeding current. To the contrary of, like, for example, in this case, when like the organism is handling its prey, it stops generating a feeding current and it's dedicating itself fully to be able to engulf this prey. Excavates will be generating a flow that goes from the anterior to the posterior end, and that's how the particles end in the are captured at the end of the cell too, and then that's where phagocytosis happen. And if you see here, you can see that there's like this kind of wave that washes through the whole ventral groove, and that is essential element to be able to engulf the prey. So that is also a way of being able to phagocytize it. If not, they would never be phagocytized. So they're characterized by this float that goes from the anterior end to the posterior end, and that's where the embedded food will end, and then it'll be phagocytized. And this is a video. You can see that the particle, sorry, will happen again. There it is. And then in the end, you can see there's something that pushes it towards the end, and that is phagocytized. So this is very characteristic from this group of flagellates that have this pocket in their cell, this ventral groove, and the veined flagellum. And lastly, just to quickly mention about prey selection, because these organisms are also able to select their prey. And this is a species that is with hairy flagella, so it has mastigonemes. And I was able to quickly see how, and this has not been published though, but how the flagellates were this flagellate was able to distinguish different strains of Synechococcus. Reasons still unknown, probably chemical cues, but there was a clear rejection of strains that, well, we colored as green, well, they were green pigmented, and pink was also quite often rejected, but the other strains were ingested in their majority. So this is another aspect of the feeding of phagotrophic flagellates that has been very, very lightly studied. And yeah, more to be known. So I've been talking about hairy flagella and then the flagella with the vein. Now let's just look at one last strategy, one more grazing strategy, and that would be the flagellar sheet. So I've talked about the typical excavates that have the vein flagellum and their ventral groove. So in the same group of excavates, we have this species or genus called Percolomonas, and they have actually 4 flagella, and you can see them beating here on this sketch here. They all beat synchronized. You have 1 long posterior flagellum and then 4 shorter ones. They all beat over the ventral groove, and you usually see them on the surface attached by the end of this long flagellum. The thing is that the fact that they're in the excavate group, but they do not have a vein. Instead, they had this weird configuration of the flagella, which kind of resembled a sheet. So we wanted to investigate a bit more about this, because knowing the advantages of having a veined flagellum with a groove, then the fact of the absence of this vein resulted a bit like, mysterious for us. So when we took a closer look, we were able to see, if you see in this first video here, how these 4 flagella are always synchronized and beating as a sheet, so they're always together. And you can also see that apart from their like longitudinal beat plane, they also have this kind of wave effect laterally. And then we're able to see that this this lateral beating was also generating like a sideway flow. So instead of having this characteristic flow that typical excavates have, that is going from the anterior to the end of their cell of the ventral groove, we have this a bit more of a lateral flow that differentiates them a bit from the strategy that we had observed before with the typical excavates or the veined flagella organisms. And then what we were also able to see is that the flow was barely observable until the particle was very, very, very close to the organism. Oh, and this video stopped. But mainly, yeah, sorry about that. But mainly you can only see particles that were very within maybe 2 micrometers of reach to the that were actually then affected by the feeding current that these organisms are producing, and then that would definitely be sucked into the ventral groove. And by seeing this, we'd also see that the flow was only produced when the 4 flagella were arching up on top of the ventral groove, meaning that when the 4 flagella are facing, are on their cycle, that they're like facing down to the ventral groove, there's no flow. So the flow was interrupted. So it's not this continuous flow that we were used to seeing in typical excavates, but it was more of a instantaneous flow that would occur when every time the 4 flagella would lift and open up the pocket, the ventral pocket. And we kind of approximated it roughly to what this like instantaneous sinking point would be, the sinking force, in which if you imagine like the force origin being where these 4 beating flagella are, the flow produced, the speed of the flow that these flagella are producing is equal on its radius and will decrease on it by the distance squared. So meaning that at 2 micrometers you can observe a high flow, but as soon as you're out more with higher radius, then the flows will be negligible, and basically it would resemble a lot of Brownian motion. So it's definitely a different pattern and a different strategy from the typical excavates that we're used to seeing. And that kind of explained why we could also observe this organism jiggling around and moving around a lot, because you can imagine that with this type of flow, it would be depleting its sources of its immediate vicinity and quite quickly. So it has, well, in a steady state, right? But yeah, it has to constantly be renewing its environment. We did some quick calculation. Well, we did some calculations of the clearance rates, and they actually also coincide more or less to the magnitude that we've calculated for other phagotrophic flagellates. And I think I have time. Yeah, I actually have time to continue a bit more. So I've talked to you about all these different strategies to generate these feeding flows, and I've talked to you about the flagellates that bear hairs on their flagella, and the flagellates that bear a vein on their flagella. And this vein is very important and acts together with this pocket or this ventral groove, and these are morphological structures or feeding structures that are characteristic from the typical excavates, right? So I want to talk a bit more about typical excavates or about excavates because this is more where we're working on now. And excavates are a group of three, well, they're this phylogenetic group that is formed by three separate groups that have always, their relationship, their phylogenetic relationship has never been very clear. And they're deep branching, meaning that they are quite primitive groups, but it has been well disputed until recently. Where do they actually belong in the eukaryotic tree of life? So here you have the eukaryotic tree of life, and then the origin where you have the LICA or the last eukaryotic common ancestor. And then you have mainly two branches that branch out from LICA. But these groups over here that are actually, some of them are quite recently, have been quite recently described. Their position within the tree has always been a bit doubtful. It's been always doubtful, and then different studies have been coming up, positioning some groups of excavates on one side and one other group on the other side. And the fact that sometimes studies would be having results of having this group divided in one on one side and the other on the other side of the tree, that was giving strong suggestions that this last eukaryotic common ancestor might have had some morphological features that these that the excavates have, namely the vein flagellum and the ventral groove. And obviously for us it's of interest too, because if they have these two features from what our studies show, then they must have fed, there's a possibility that the last eukaryotic common ancestor, this eukaryotic cell that we all descend from, might have fed the way we're observing these typical excavates doing so. So, and it's just been recently, so this year, where this last study that I'm citing here has found stronger evidence that yes, we can find this excavate group, which is here in red, divided in one on each side of the tree, of the two branches of the eukaryotic tree of life. And given the database that they used and so on, and the fact that these groups have the features that I've mentioned before, the morphological features I've mentioned before, then there are suggesting that this is really strong evidence that this primitive eukaryotic cell that we all descend from have actually these morphological features too. Yeah, so we have typical excavates, and I've already talked about why they have been sparking more interest recently. And then because of like their morphology, in which they have this posterior flagellum in blue with a yellow, with the vein that's represented in yellow, and the ventral groove. Well, I should also mention that we have some excavate-like flagellates, because within this group, all these up until E do belong to excavate groups, but F and G would be the imposters, as we could say. Imposters because they do not belong to the phylogenetically to any excavate group. They actually are outside. One is like a probota here, and the other one is an alveolate, so it belongs to the side. So what we're seeing here is that, yes, excavates are on each side of the tree, but then we also have some other organisms with the same features that are outside of the excavate group. Then there's two questions here. First, how are they using these feeding structures? Because we've well defined how the typical excavates are using them, but are these organisms also using them in the same way, producing flows to be able to capture their food? And then the second question is, what are these? How have these organisms evolved to have these structures? Whether the origin is because they share a common ancestor in which they have they have obtained these features, or whether these these structures have evolved independently. So the thing is that actually these flagellates that I've mentioned that have the same feeding structures don't feed on bacteria or phytoplankton. They actually feed on other flagellates. So here you have a predator that is going to capture another flagellate. And as you can see, or I don't know if you've been able to perceive it, but there's not actually a flow. The feeding mechanisms that I have been showing you until now are definitely different from what we can see here on this video. So we're seeing that these organisms are feeding on a different type of food and have a different mechanism on preying on their prey, and that is completely independent of generating a feeding current. So more than generating feeding flows, they're more like crawling around the surface and searching for their food, and apparently it's more of a random encounter, and so we're like denominating them more like crawling hunters. We can see that their behavior has changes depending if they have prey or if they don't have prey. So these are tracks of them, and sorry, I think these are in. Oh yeah, no, it's already changed in micrometers. But we can see that in the absence of prey, or in the almost depletion of prey, their swimming tracks are much more active, and their displacement here, as you can see, is much more greater than when they are in the presence. They have plenty of food. So their behavior for searching for food changes. And also the amount of them free swimming also changes depending if there's food or no food. So they'll tend to attach to surfaces when there's a lot of food, but then they'll tend to free swim and potentially look for new patches of food sources when the availability is low. Also, what we were able to observe is that their flagella has nothing to do with handling their food. I showed you before that, that apart from generating flow, the flagella can also be useful to be able to handle the flow, the food, and to be able to ingest it posteriorly. But in this case, we were able to observe that the flagella have no role at all in being able to capture their food. So as long as there's a contact, and then they're able to attach to each other, and the flagella have no role at all in the handling of it. So this leaves us to a question, the last question also, of like whether this these morphological features, how have they evolved? Either it can be an homoplasy, which, as like in the case of the wing of a bird and a bat, they have evolved independently, serving for the same function, or we are more inclining towards the possibility of it being an homology, so an homologous trait, basically that these characters of the ventral groove and the vein flagellum originate also in the same manner as how the typical excavates have. So maybe giving a stronger indication that the last eukaryotic common ancestor, so this original eukaryotic cell at the root of the eukaryotic tree of life, had these characters, and then both groups have evolved with them, and their functions have then diversified. But this is still an open question, of course. And that's about it. Yeah, just as a quick summary, we have been able to see that these phagotrophic flagellates have many ways of feeding, and it's a result of their different morphologies, and they're also very diverse phylogenetically. I hope you've been able to have a good overview of these these three ways of feeding. You have the hairs, you have the veins, and then you have the flagella sheets. So the flagella is never a simple component, and it has and it's a very versatile structure that serves for the flagellates in many ways, not only for feeding but also for displacement, and then also for sensory functions. And then, yeah, we were able to see that all of them, despite of their diversity of mechanisms of generating these feeding currents, they all produce an expected magnitude of clearances that are, well, yeah, have been calculated in the past for that are like necessary magnitudes for their survival. And then studying these newer flagellates that are preying on other flagellates, we kind of see that the fact that they have a different function that might be giving stronger evidence that Lika was more like a typical excavator and might have been feeding like one. So, and that's about it. Thank you very much.
Fantastic. Thank you so much, Sei, for this fascinating talk and work. Really, really good. We have plenty of time for questions. So if you have questions in the, yeah, please feel free to ask your questions or Marco.
Yeah, hi, Sei. Very nice talk. So about the mastigonyms, can you hear me?
Yeah.
Okay. So I had a question about mastigonyms. So the mastigonyms that you have been referring to are quite stiff, right? But not all microorganisms that have mastigonyms have stiff mastigonyms. So for example, clammy has more floppy.
Yeah.
Mastigonyms have a slightly different structure, I guess. So I was wondering whether there are, I mean, how prevalent are these other types of mastigonyms? Whether you know anything about that, and if you have encountered any species where the mastigonyms are of this type, and what type of feeding would they be displaying?
So my knowledge on other types of mastigonyms is very minimum. On the case of Chlamydomonas, I don't recall how they use them, but so mastigonyms are not only generating more efficiently this feeding flow, but in my sense, from the observations I have, is that they also are a sensory or structure. So I can imagine that these more floppy hairs are doing more of that function, although I've never observed it. I do know that some of these organisms that I'm seeing now that prey on other flagellates, I do see that some of them bear mastigonyms too, but only like in the beginning at the proximal end of their flagellum, so the one that's
proximal
closer
or the
to.
distal?
Sorry?
The proximal or the distal?
Proximal.
So towards the cell.
Yes.
Okay.
I've seen some images, some electronics microscopy images of that. So yeah, that's when my sense goes towards more that they are doing more of a sensory function than definitely not producing the flow.
Okay.
Because these organisms have like a bit of they've been able to observe these hairs in the proximal side, and then they have this more or less obvious vein. So they're actually bearing both of these structures.
Okay, thanks.
But I can't confirm if these hairs are stiff or not. That's what I don't know.
Okay, thank you.
You're welcome.
Okay, we can take a question from the chat as well. Marcela Castaño says that the microphone is not working, so I'm gonna ask this question. She said, "Thank you for the talk. It was very interesting. I particularly like the video clips. I was curious if there is any particular behavior of the flagella when they reject a potential food particle, or even the excavates, like maybe they don't use to capture or for rejecting it. Just a curious question.
Yeah, and that's a very good one. And I actually wasn't able to talk about it, but okay, so first talking about the excavates, rejection wise, it was not much of a obvious change of behavior in their flagella. Maybe they stop a bit, but that's about it. Sorry. The limitation or like the food selection for them is more physical in the sense that how much can fit inside their ventral groove, because that is the food has to be able to enter the ventral groove to be able to be phagocytized. So it was the size of the ventral groove that is selecting prey whether to be eaten or not. And then maybe there has been a bit, you can see some food just pop out because the feeding current obviously is going from the anterior to the posterior end of the pocket. So if the flagellate was too full, say that, then maybe the phagocytosis won't occur, and then the particles in the end will just be like washed away with the flow, to say it in a general way. But the flagellates that bear the hairs, I was able to observe many different rejection behaviors. And particularly, there was one species, the one that has this very long flagellum that curls around. It's called pseudobodo. It was able to capture its prey, and when it captured prey, it would like change a bit the configuration and start beating very fast to be able to retain the prey. And in that sense, and in that moment, if it was ingested, then phagocytosis would occur. But if not, it would just return to its original position. So this change of configuration from capture to just generating feeding flow would occur, and then the particle would be embedded again into this feeding flow, and it'd be rejected. But then the same species sometimes would be able to completely like untangle its long flagellum and actually physically push away the prey when it decided to not, that it didn't want to eat it. And sometimes that happened even not even capturing it. It would sense, and especially it happened with bigger particles that were approaching the predator. Then suddenly it would just change its configuration and physically try to push away the particle. So yeah, there is many different ways of rejecting a prey.
Thank you, Sei. Oscar has a question.
Hi, Sei. Can you hear me?
Yeah, hi.
Hi. Very nice talk. Thank you. I was wondering about what you mentioned at the end of the talk, that flagella can fulfill different functions, just feeding, but also locomotion and mechanosensing, no? So I'm guessing that the species with which you have worked are mostly sessile, or they don't move too much. But I was wondering if they can also use the flagella for moving. And also, I'm wondering whether you have a sense of what sort of trade-offs are involved. So is any of these structures, these hairs or the veins, are capable of fulfilling other functions in the life of flagella?
So yeah, I mean, well, first of all, with the sessile, well, these organisms, yes, maybe I've not emphasized it too much enough, or I didn't mention it properly. They feed, well, obviously there's a bias of me being able to observe them more easily when they're attached to the surface, and most of them do. But then, yes, they will be using the flagella to swim away and find other locations. Sometimes the way they're beating their flagella will change. Sometimes there's some species that will change totally of phase or yeah, of morphologically will change to be able to be more of a motile morphology, to be able to swim more efficiently. So yeah, the flagella is the way I've explained to you is when they're attached and generating the feeding current, but then they will use it in this. They will use the flagella in the same way, or maybe in a different different behavior to be able to swim. Some are more efficient than others. As a trade-off, it is known that, and going back to what's the swimming, it is known that they're able to produce more force swimming than not attached. So they would be more efficient capturing prey while swimming. But seeing that they attach mostly to be able to feed, we can understand that that behavior is due to the abundance of food sources on surfaces that you can find in the natural environments, right? Like marine snow, or even carcasses of big phytoplankton, or any other surface you can imagine, right? And then about the trade-off of these feeding structures when wanting to swim, if I remember right, or any trade-off that these or other functions that they must have. Well, the mastigonemes I've mentioned about me thinking, well, I believe that they have also a mechanosensing function, but I cannot think of any negative trade-off. I mean, if anything, and we've not looked into this, but we don't know the role of this vein flagellum and the ventral groove for locomotion. So that would be maybe something, and that's something that we're trying to look into currently. But yeah, that's more or less. I think I've gone over the two questions.
Yes, thank you very much.
Thank you.
Thank you. Are there more questions in the chat or in the group? I had a similar question, say, but I think you already answered about this. Do you think that they can use this maybe to form clusters as well if they are in the natural environments?
To use what?
To use this flagella or this appendages to form clusters?
Ah, no. No. They, and I only know from my observations from cultures, right? But in the lab. But if they form clusters, is not actually clusters, it's more that they're all attached to the same particles and to the same particle, and they're all like feeding from the bulk from the same particle, but they're never attached flagella to flagella. Is that what you mean?
Yeah.
Yeah.
Or to bodies. Yeah.
No. The flagella, a lot of flagella end within their distal tip with this thinner ending that is a bit structurally different, if I recall right, and it's called an acronym. And I think they kind of relate the function of this distal tip to attaching. But I've not heard of, or at least the flagellates that I observe are individual in the matter. Yeah. I do know of others that do form some sort of colony, but I don't think it's through the flagella attachment. I think it's more of some other structures, and they're like residing inside some kind of like loricas and so on. But maybe somebody knows and can correct me.
Thank you. Are there more questions? We have 4 more minutes. I'm tempted to ask another question, but I know it's at the edge of your work, but so it's regarding LICA. So you know how much I like LICA. So so this means that LICA was a complex cell, which is not very surprising, that has probably typical excavate behavior or flagellum. Do you think it has some appendage or flagellum that helped LICA feed on mostly bacteria, or something that could do the same behavior as this one, feeding on other also LICA-like population, or being like or other similar eukaryotic cells?
I think that there is a majority of a consensus that LICA was fed on bacteria. I am quite sure of that. So yeah, it would be more on bacteria than on other flagellates. That is what has been told about.
Makes sense.
Yeah. Well,
Thank
yeah, we're still investigating it, right? But the function of this veined flagellum and the ventral groove in these eukaryovorous, or these flagellates that feed on other flagellates, the function of these feeding structures are still quite a mystery, in the sense that the way they capture their food is through these other organelles that they have that basically they shoot at their prey to be able to stun them and to attach to them. Still ongoing research, but yeah. And there is some evidence in the literature that, like, the people that have described these species and that have done more ultrastructural work on them. Yeah, but it's very, very different. So the fact that we don't know the function of this vein and ventral groove in these type of flagellates would probably also indicate the fact that LICA was bacteriurous, so fed on bacteria.
Thank you. I think we're on the hour, unless there is a last minute question. Other than that, I would like to thank you, Sei. Again, thank you so much for this fascinating talk and research. And we will see you probably at the Microscale Ocean Biophysics Conference again. Thank you so much.
Thank you very much.